Another bunch that enter at the level of succinyl coenzyme A, isoleucine, methionine, threonine, and valine. A couple that enter at the level of fumarate. And then we did talk about the ones that enter at the level of oxaloacetate, and that's aspartate and asparagine. Then a bunch enter at pyruvate. We talked specifically about alanine, but these others also enter there. Now, those are colored purple. And the reason they're colored purple is that they all feed into either the citric acid cycle intermediates themselves or into pyruvate. And all of these molecules are able to synthesize glucose. And the way we do that, if you feed into pyruvate, we then convert that to oxaloacetate, as I showed over here, then the phosphoenolpyruvate, and then we undergo gluconeogenesis up to glucose. So all of these molecules here are called glucogenic. All of these amino acids feed into the ability to synthesize glucose, so they are glucogenic. The yellow ones are not going to be glucogenic because they feed into acetyl coenzyme A or into acetoacetyl-CoA, which is converted later into acetyl-CoA. For every molecule of acetyl-CoA, we completely break that molecule up and release the carbons as CO2s. So we can't generate, in a net sense, glucose from acetyl-CoA. We'll find in the next chapter that that is also true of the fatty acids. They all are metabolized to acetyl-CoA. We can't synthesize carbohydrates from fatty acids, okay? And so this separates the amino acids very nice into the glucogenic amino acids that feed into these citric acid cycle intermediates and what are called the ketogenic amino acids that feed into acetyl coenzyme A. So we've looked at several of these: alanine, glutamate, glutamine, aspartate, asparagine, but we have not looked at all of these, and as I mentioned, we're not going to cover the metabolic pathways of all 20 of these, but this just gives you an idea of how all of them feed into the cycle. Yeah?
There are a couple of those that overlap, so that just means that they can do either one depending on how they...
That's right. I have another slide that shows that. The ones that overlap can both form acetyl-CoA and make what are called ketone bodies that we'll get into in the chapter on the fatty acids. And they can be gluconeogenic, as you can see here. So the tyrosine there, part of the framework of the tyrosine enters as acetoacetyl-CoA, but part of it also enters at the level of fumarate. So some of them can do both. You may have heard of the Atkins diet, which is high in fats and proteins and low in carbohydrates. I'm not quite sure how it works because you can see most of the amino acids are able to synthesize carbohydrates. So most of our protein can be used to synthesize carbohydrates. What you're trying to do in the Atkins diet is shut down the citric acid cycle so that you metabolize the fat more rapidly to keep up with the needs of metabolism. But having a lot of these kinds of amino acids anyways is going to fill up the citric acid cycle. Here is that table that I was just saying. These amino acids are what we call glucogenic. These ones here are purely ketogenic, and these are both, okay? So leucine and lysine, they would not at all support the synthesis of glucose. These others would to varying extent.
What does ketogenic mean?
It's a term that we'll get into in a later chapter, but it means the ability to form what are called ketone bodies, which are generated during a metabolic condition called ketosis, which is, happens with diabetic patients and other metabolic conditions. It's basically a condition in which the citric acid cycle is depleted of all the intermediates and shuts down. So the ketosis is when we shut down the citric acid cycle. Okay? Now, this is the cycle I was referring to. We have amino acid metabolism in all of the cells of the body, but excess ammonia is finally shuttled into the liver. An example of this shuttle is shown here. In the muscle cell, we undergo glycolysis down to pyruvate. Ammonia groups from all of the amino acids are transferred to form alanine. And again, this is this transamination reaction. We start with pyruvate, do a transamination reaction. The ammonia is incorporated into the alanine. The alanine is then sent to the bloodstream where it travels to the liver. The alanine then undergoes a transamination reaction to glutamate. The glutamate now carries the ammonia. It then releases free ammonia, and then we enter the urea cycle that we just talked about. The pyruvate that results from this undergoes gluconeogenesis back up to glucose and completes the cycle. So the net purpose of this cycle is to transfer excess ammonia ions into the liver cell where they can be converted into urea and then ultimately sent to the kidneys for excretion. Okay? So that's the transfer, it's called the alanine cycle. Okay, this is just the printed version of the urea cycle here. And we've gone through this all on the board here, so I won't do that again, but this is the full thing of the urea cycle. The conversion of CO2 and ammonia into the carbomoyl phosphate using 2 ATPs. The combination of ornithine with the carbomoyl phosphate to form citrulline. The combination of aspartate with that to form arginosuccinate. The formation of arginine and fumarate. And then the release of urea to get back to the starting point. This picture also shows this little part of the citric acid cycle where we convert fumarate to oxaloacetate, and then by a combination of transamination reactions and reductive amination, we reform the aspartic acid. So that shows everything, the urea cycle plus that part of the citric acid cycle which is used. Okay, here are the summary equations. I've already had these on the board, but the urea cycle proper involves aspartate converted to fumarate in the formation of urea. If we do that extra part of the citric acid cycle, this is the summary directly forming nothing but urea and the fragments of ATP. Okay? So, now I'd like to do just a little bit more, we're going to switch now to the next chapter, 31, and I'm going to talk a little bit about biosynthesis. Now, a lot of biosynthesis we've already covered. The transamination reaction is reversible. The oxidative deamination reaction is reversible. So we can just reverse a lot of what we talked about for the synthesis of the amino acids, at least the common ones that we talked about. Now, as you know, as humans, there are certain amino acids we have to have in our diet. We are not able to synthesize these amino acids ourselves, and we call these essential amino acids. There's other amino acids that are non-essential because we are able to synthesize them. They tend to be the smaller and simpler ones that can be synthesized in a smaller number of steps. There's a couple of these that are kind of ambiguous, the arginine and the histidine. It says here that the histidine is needed for children, but not necessarily for adults. Arginine can be synthesized in mammals, but not very efficiently, so we really need extra arginine in our diets. So those are essential and non-essential amino acids. What I want to do now is just kind of go over a few of the biosynthetic pathways to give some general principles. We've already talked about this. In this direction, this is the oxidative deamination, converting glutamate to alpha-ketoglutarate and ammonia. In the synthesis direction, it's called a reductive amination. We combine the ammonia with alpha-ketoglutarate, reducing power to make glutamate and the oxidized coenzyme. This indicates here that we can use either NADH as the reducing agent or NADPH. And that depends to a large extent on where this occurs within the cell. The NADPH is largely found in the cytoplasm, whereas the NADH is largely found in the mitochondria. So certain enzymes that carry out this synthesis use the NADPH rather than the NADH if the synthesis occurs out in the cytoplasm. So there we've made our glutamate. So that's one of the reactions. Here is a interesting reaction. We've already talked about glutamine and asparagine. These are amino acids that have their terminal carboxyl group in an amide bond with an extra nitrogen. Here we take glutamate, and that's the terminal side chain carboxyl group. We first activate it with ATP to form a phosphate intermediate. Then we react it with ammonia, using the energy of that bond right there to form the glutamine. And so this has that amide bond with a carbonyl oxygen and an NH2. So that is the synthetic pathway for synthesizing glutamine, a very simple sort of pathway. Now, what's particularly interesting about this reaction is that it is highly regulated. And that's because the glutamine is the source of ammonia for all sorts of other molecules. And I'll just show this in a slide here. This is a bit of a mess, but it gives you an idea of how many different molecules get their ammonia from the glutamine. So here's the glutamate. There's the reaction in which we form the glutamine down here, okay? Then all of these other molecules get their amino groups from the glutamine. The carbomoyl phosphate we've already talked about, the serine amino acid, histidine, tryptophan. Here's a glucosamine 6-phosphate. Here's a nucleotide, CTP, etc. All sorts of stuff. What's interesting about this is that this is one of the most complex and interesting forms of feedback inhibition that is known. We don't want this reaction occurring full speed all the time, but since so many molecules are derived from glutamine, we have to have a complicated feedback inhibition system. So all of these end products are inhibitors of the reaction itself, in addition to glycine and alanine, as you see up here. So this enzyme, which is a very large enzyme of 6 subunits, it has binding sites for all of these molecules here that are separate. And each one has its own inhibitory effect on that enzyme. And it's kind of a combination inhibition type of pattern. They all inhibit a little bit. To totally shut down the enzyme, they all have to be present. If there's just a few of them present, it'll be partially inhibited, but it'll still go to provide glutamine for the ones that are still at low amounts. So a very interesting example of feedback inhibition. Okay, I've got just a couple more of metabolic stuff. This is a flow chart. We're not going to go through all of the details of this, but this just indicates how all of the amino acids are synthesized. And a lot of them, as you can see, come from the citric acid cycle, asparagine, methionine, threonine, leucine, through oxaloacetate, through the alpha-ketoglutarate, we get all of these guys. But then glycolysis produces some of these and so forth. So that's just kind of an overall roadmap. That's a picture that's shown in your textbook. Here then is just a couple others that I wanted to talk about. This is the serine synthesis, and it comes from a glycolysis intermediate, 3-phosphoglycerate that we found. It is first oxidized to this molecule of phosphohydroxypyruvate. Then it is converted by a transamination reaction with glutamate to phosphoserine, and finally it becomes serine. So there's a three-step synthesis that results in the synthesis of serine. So that's another example of one of the amino acids that has a, and that's a non-essential amino acid. We're fully capable of doing that. Now, I wanted to just give you a flavor of another general reaction, and this is how we transfer a methyl group. Methyl group transfer is very important in assembling the carbon framework of a lot of different molecules. And the tetrahydrofolate coenzyme is the coenzyme involved in transferring methyl groups of various types. We've encountered a lot of different coenzymes. Well, here's another one, tetrahydrofolate. And it reacts, we've already synthesized serine, that's why I decided to study serine. If we react serine with tetrahydrofolate, we transfer one of the carbons of the serine to form this molecule of N5-N10 methylene tetrahydrofolate. The carbon in serine is right there, okay? So this is a methylated form of that tetrahydrofolate, that special coenzyme for transferring methyl groups. That can be reduced to form just a N5 a pure methyl rather than a methylene using reducing power. This just shows the reaction stoichiometry again. The serine plus THF goes to lysine plus the methylene THF. The glycine then can be further degraded into CO2 and ammonia, transferring its central carbon atom to the THF to form, again, the methylene THF. So this is another example. We can synthesize serine by the previous slide. We can then synthesize glycine using the THF to receive the methyl group. Then if we want to break down the glycine, we can break that down into CO2 and ammonia, transferring that central methyl of the glycine into this methylene THF. So the idea here is that tetrahydrofolate is kind of an important methyl group transfer. There's one other molecule that's very important in methyl group transfer, and that's the adenosine right here. It's called SAM for short, S-adenosylmethionine. This is kind of complicated, but we've already covered some of it. Let's start right here. Here is homocysteine, okay? And homocysteine is just like cysteine, but it's got one more methylene group. So it's an amino acid. There's the alpha, there's the carboxyl group, the amino group, two methylenes, and a SH group. What we do next is we bring in this methyl THF that we talked about in the previous slide. We transfer that methyl group to the homocysteine to make the amino acid methionine. And here's its methyl. So that's how we synthesize methionine. We then use some energy of ATP, and we take the adenosyl group of the ATP and attach that to the methionine to form this S-adenosylmethionine, or SAM for short. And what's important about this is the methyl group that came from the THF here is now that methyl group on that adenosylmethionine. That plays a central group and methyl group a central role in methyl group transfer to all sorts of metabolites. That methyl group is transferred to a methyl acceptor to make a methylated acceptor. That makes this molecule here that's the same as that one but missing the methyl group. It's called adenosyl homocysteine. Then we break off this and we get back to the homocysteine. So that just shows you in general terms how we carry out this methyl group transfer. It's a combination of the tetrahydrofolate and the methionine and the SAM molecule. You see here we've got this little cycle we already talked about. The THF can accept one of the carbons from glycine to form this. That is then reduced into the methyl form of the THF to allow that to occur. So that's the story on how we do this methyl group transfer. Any questions on that? And again, I'm not going through all the amino acids, but so we've just sort of focused here on a few of the amino acids in the general procedure for transferring these methyl groups. Okay, I've got just one more topic left, and this is the topic of how we get nitrogen in the first place into the biosphere. These days, a lot of our nitrogen comes from fertilizer in the so-called Haber-Bosch process, and that takes an enormous amount of energy. It turns out that the nitrogen molecule is very, very stable. So, I've got any spare blackboards left. There we go. So, what we're talking about here is this molecule of nitrogen, N2, is very, very stable. If we want to react this with hydrogen, it takes an enormous amount of energy to do that. And formally, we can do that by taking this molecule of N2 and reacting that with 3 molecules of hydrogen, and that's going to generate 2 molecules of ammonia. That can be done in what is called the Haber process. It takes an enormous amount of energy. It takes an enormous heat, very high atmospheres, very complicated process. Just to give you some idea, it occurs at 500 degrees centigrade. It occurs at 300 atmospheres of pressure. It requires a very complicated iron catalyst to carry out. It's enormously costly energetically. A lot of our energy is used to make fertilizer by this Haber process. However, as you know, certain organisms, certain bacteria called the ribosomes in the root nodules of legume plants are able to carry out this nitrogen fixation process under very mild conditions. And this just shows here an outline of that process. And you can see here that these denitrification bacteria are able to form this ammonia molecule. This shows some other things involving nitrate as well, the conversion of ammonia into nitrate, which is also a form of fertilizer. But we're going to really focus on how this process is carried out by these particular bacteria. And these are the rhizobium type of bacteria. So, this is just a picture of these root nodules where the bacteria are living in a symbiotic relationship with the legume plants like clover and alfalfa. And here is a scheme of the overall process. What is occurring here is that we need electrons that derive from ferredoxin, which is an iron-sulfur protein with a very negative redox potential. The nitrogenase enzyme that carries out this reaction has 2 components, a reductase component that's also called the iron protein, and the nitrogenase itself that is composed of both molybdenum and iron. The net reaction that is carried out is shown here. 1 molecule of nitrogen, a molecule, N2, reacts with 8 electrons, 8 protons, 16 ATP. We make 2 ammonias, 1 hydrogen, and we bust up all the ATPs. So you can see that from the standpoint of the bacteria, this takes an enormous amount of energy. It's also a process that has to take place under strictly anaerobic conditions. So these root nodules have specific systems to remove all of the oxygen. the oxygen will poison the system. And you can see here by the fact that there's 16 ATPs involved means that we need a lot of energy to carry out this reaction. And what all that ATP is doing is boosting the redox potentials of those electrons more negative so that it can carry out the reduction of N2 to ammonia. That's an enormously negative redox potential, an enormously difficult reaction. We need all those ATP molecules to boost the redox potential to more negative values to drive that reduction of N2 to ammonia. And in recent years, people have done the complete X-ray crystal structure of this molecule. This just gives you a little flavor of what's going on. Here is that first protein, the reductase. It has a binding site for the iron sulfur where the electrons are transferred and binding sites for the ATP. The ATP binds, is hydrolyzed, and changes the conformation of this iron sulfur cluster, which is highlighted here, and it changes in a way to make its redox potential more negative, so it gives enough push to those electrons to carry out that reduction of N2 to ammonia. Here is the other part of the enzyme, the molybdenum iron part. Here is a blow-up of the part that includes molybdenum, and all the iron atoms are colored kind of green here. The molybdenum is right there. The yellow things are sulfurs. We've got a peak cluster that's just iron and sulfur. We have the cofactor here that's got molybdenum and iron. Right in there, that's where that nitrogen is going to be reduced to the ammonia. That's the active site right there, and there's still a lot of active research being carried out on exactly how that does that reaction, because it's so important to our agricultural system. But somehow the electrons come in here, they're transferred to there, and then that nitrogen is reduced to ammonia. And this last picture is just a hypothetical mechanism of how this works. This is still an active research area. We start with a metal, and this is actually thought to be an iron next to the molybdenum. The nitrogen molecule binds to it. That nitrogen is highly reduced. It transfers the first electron and a proton to make this thing, another electron, another proton to make this molecule. Now you can see we're beginning to reduce it. Then we release that one ammonia. We're left with one nitrogen left. We reduce it again and again, adding more protons, and finally we release the ammonia at the end. So something like this is how this nitrogenase enzyme works. But obviously it's a very important reaction in the biosphere to carry out this formation of ammonia that we're all dependent on. We can't ourselves use ordinary N2. Most plants can't use ordinary N2. It's only these rhizobium bacteria that are able to fix this N2 into the useful form, which is ammonia. Okay, any questions on that? Okay, well, we'll see you then on Thursday.
Early.